Hello and welcome everyone to this first screencast lecture on muscles and muscle tissue. Before we get started with this screencast, if I haven't had a chance to mention it in class already, I'd like to go over the format of the way I'm going to be covering each of the different systems in the human body. So whenever we start out, you're going to see usually the first couple slides or first slide or so I'm going to have information I call either overview or introduction information, giving you some background, talking about importance, answering questions oftentimes that students have, FYI information, so on and so forth. Once we cover that background information or that overview information, we'll move on then to talk about the functions of the system. Why is that system there? Why do we need to discuss it? After we complete functions, then we'll start to take a look at the anatomy of the system. We'll take a look at the component parts, what's there, and we'll talk about why those parts are there. And then we'll talk about the physiology of the system. We'll talk about answering the question, how does what's there do what it does? You know, we're in an anatomy and physiology class, and anatomy and physiology really attempts to answer three questions about each of the different systems in the human body. Those three questions are, what's there, what does what's there do, and how does it do what it does is the third question. That's the physiology question. So my PowerPoints are set up to give you the overview, talk about why we have that system, and then answer those first two questions, what's there and what does it do? That's the anatomy component of it. And then the third question is, how does it do what it does? That's the physiology component. Once we discuss the physiology component, then we'll always kind of finish up by talking a little bit about some practical applications in healthcare. How would you use this information clinically? Okay, so let's go ahead and get started with our first slide. And our first slide is an overview slide. And in terms of background, one of the things you may recall from A&P 1 is that muscle tissue is one of the four primary tissues of the body. Can you think of the other three? Elastic tissue, connective tissue, and nervous tissue. Those three, along with muscle tissue, make up the four primary tissues of the human body. So here, we're going to be talking extensively about muscle tissue, its structure, function, and physiology. Now, whenever we take a look at muscle tissue, we understand that muscle tissue accounts for almost half of your total body weight. So it can make a big difference in your body weight by developing or having a loss of muscle tissue. Muscle tissue is the most dominant tissue of your heart and of other visceral organs. Remember, visceral organs, you may have been taught in other classes, are those organs that are inside your body. So organs that function entirely and are located entirely inside the body, particularly in the abdominopelvic cavity, are all considered to be visceral organs. Muscle tissue is the dominant tissue of the heart, and which is a visceral organ, and other visceral organs in the body. All right, let's take a look at the properties of muscle tissue. When we look at properties of muscle tissue, we're really talking about function. What does muscle tissue do, or what does it have the ability to do? Well, our first property, and this is the most important property muscle tissue has, is contractility. For now, you notice that I have an asterisk here in front of contractility. For now, we're going to say that contractility is going to be defined as the ability of a muscle tissue or muscle to shorten. Now, that definition will be evolving as we go along, but for now, in the beginning aspect, we'll take a look at it from the standpoint of its ability to shorten. But in addition to contractility, muscle tissue has other properties as well. For example, it's extensible. We say extensibility is another property of muscle tissue. Extensibility is the ability to stretch. So muscle tissue, let's say, for example, like, can you see this rubber band that I have here? Where do I want to put it up? Right there. So you take a look at this rubber band, also called an elastic band. One of the things we know is the property of this is it has the ability to stretch like this. I can stretch it out. So the rubber band is extensible. Muscle tissue can be stretched just like this rubber band can, so it also has that property of being extensible. Now, once I stretch it out, if I let one side go, we can see that it kind of snaps back to its original shape and position. That's another property which is referred to as elasticity. So this rubber band has the property of elasticity, the ability to return to its original length after it's being, in this case with the rubber band, after it's being stretched, it has the ability to snap back. For muscle tissue, elasticity refers to the ability not only to snap back after it's being extended this way, but to also return to its original position or original length after a contraction takes place. So we'll be looking at that as we go along as well. And finally, muscle tissue also has the property of irritability. Now, that doesn't mean it gets up on the wrong side of the bed every morning, but what it does mean is that in terms of physiology, irritability means that a tissue has the ability to respond to some kind of environmental change, or what we refer to as a stimulus. Okay, so muscles do have the ability to respond to a stimulus. How do they respond to that stimulus? By undergoing a contraction. All right, now let's take a look at some more specific functions of, let's say, the muscular system in general. Obviously, our muscular system is that we're going to be talking about here is primarily going to be focused on muscles that are attached to the skeleton of the body. And because those muscles are attached to the skeleton of the body, they're going to help to maintain posture. They can help me to sit upright, they can help me to stand, not slouch my shoulders, so on and so forth. But in addition to just maintaining posture, muscles are also going to help to stabilize our joints. Now, we usually think of joint stabilization as being a job of the ligaments. Ligaments are bands of connective tissue that connect bones to bones. But there's additional support that's provided around joints by the fact that oftentimes muscle tendons cross the joints and attach to bones on different sides of the joint. This helps to stabilize the joint. We'll be seeing that as we go along as well. Muscles also are important because they generate heat. That heat is important to helping to maintain what we call thermostatic homeostasis, maintain the proper balance of heat in the body so that cells can work most effectively. We'll be taking a look at how muscles do generate heat as we go along. You might be surprised at the answer to how they do that. And of course, if I were to ask you the question, what's a function of muscle? This is the answer you might come up with first. Their job is to produce movement. Because they are attached to the skeleton, when muscles undergo contraction, they pull on bones, and when the bones are pulled, they move, and that helps us to move around through our environment. So these are all functions of muscles, and therefore of muscle tissues as well. So you may have done this already in A&P1, but I want to refocus on this so that we bring this back and place this plate back on the table about the types of muscle tissue, so it's first and foremost in our minds as we continue to go on through this PowerPoint. So we take a look at the types of muscle tissue, and we find there are three types, you may recall, of muscle tissue: smooth, skeletal, and cardiac muscle tissue. Let's do a quick review of each one of these three different types of muscle tissue. The first type we'll take a look at is smooth muscle tissue. Smooth muscle tissue is the type of muscle tissue that's found in the walls of all the visceral organs other than the heart. Remember, those visceral organs are the ones that are inside our body. That would include things like blood vessels, the stomach, the intestines, so on and so forth. All those visceral organs. Okay. Next up, we take a look at smooth muscle tissue. We look at the cells of smooth muscle tissue, and we refer to the cells of muscle as fibers, because when muscle fibers were, when muscle cells were first discovered, we have no idea they were cells, and they look like little threads or fibers of material, and so they were originally referred to as fibers, and all this time later that name is stuck. So when we refer to muscle fibers, we're referring to the types of cells that make up the muscle tissue. So when we take a look at the cells of smooth muscle tissue, we find they're spindle-shaped cells or fibers. If you take a look here at the top right-hand diagram, this is an artist's conception of a typical smooth muscle fiber, and you can see that the smooth muscle fiber is pointed at both ends and kind of thick in the middle. That's characteristic of a spindle. Now, what you may also notice here is there is a nucleus. The nucleus is that purple oval in the center of the muscle fiber. And smooth muscle fibers tend to have a single nucleus, and that single nucleus, just as is demonstrated here, is centrally located in the muscle fiber. One of the reasons why smooth muscle tissue gets its name is because it lacks striations. It lacks kind of cross-banding. We'll get a better idea of what cross-banding is a little bit later on. It's hard for me to describe it here because there is no cross-banding in smooth muscle tissue, or there are no striations in smooth muscle tissue. So it looks smooth and it looks the same across the tissue and across the individual muscle fibers as well. So no striations. And finally, smooth muscle tissue is involuntary. Whenever we talk about involuntary, it means that it's not under conscious control. Smooth muscle does not require us to think about it in order for it to undergo contraction. It is stimulated by the autonomic nervous system, and it automatically contracts at the appropriate time. Just before we leave this slide, you can see over here that we have, this is a micrograph of smooth muscle tissue and what it looks like underneath the microscope. And so it's a little bit difficult to see individual muscle fibers here, but you can see nuclei and you can see the smooth, consistent nature of the cells as you move across the tissue from one side to the other. Down here in this bottom diagram, we can see some individual muscle fibers that have been teased out, and this helps us to really kind of see the spindle nature of the smooth muscle fibers. So I wanted to point those out. Okay, let's move on to our next type of muscle tissue, which is cardiac muscle tissue. Cardiac muscle tissue, as its name indicates, is the type of tissue that makes up the walls of the heart. So when we take a look at this term cardiac, maybe some of you have had medical terminology already. In medical terminology, the suffix AC means pertaining to. And of course, cardi is the root word that comes from the German. It means heart. So literally pertaining to the heart. So cardiac muscle tissue is the type of muscle tissue that makes up the walls of the heart. We take a look at cardiac muscle tissue and we focus on the fibers. We see that the fibers are branched and that they are striated. So we can see the branched nature. Here's an artist's conception of a typical muscle fiber. And we can see on either end the muscle fiber branches. Sometimes that's referred to as bifurcations. We say that the muscle fibers are bifurcated on their ends. And we also note that they are striated. Now we can very plainly see as we move across this muscle fiber the various striations, the crossbands that there are. Next up, we take a look at the nucleus, and we can see that the nucleus, again, just like smooth muscle tissue, is centrally located, and there's a single nucleus in each muscle fiber. Now, the muscle fibers in the heart tend to be kind of almost, we might say, stitched together. They're fused together, and the sites where they are fused together are referred to as intercalated discs. These intercalated discs represent the fusion of adjacent membranes. Intercalated discs are actually desmosomes that you may recall learning about in AMP1. Desmosomes are one of several ways that cells in membranes can be stitched together. So we can see here on our actual, this is a micrograph of muscle tissue. We can see an individual muscle fiber right here, and we can see these kind of light lines on either end of the muscle fiber. These are areas of intercalated discs. So there's an intercalated disc there, there's an intercalated disc there. We can see some other areas where it's a little more difficult, but there's an intercalated disc there. And we can also, by the way, see the branched nature of these fibers. That aids with the spreading of impulses across the fibers to get the heart to contract in a way that it's supposed to maximize pumping of blood. Cardiac muscle tissue, like smooth muscle tissue, is also involuntary, so we don't have to think about it. Thank goodness. You know, you're taking an exam, you're working out, you're doing whatever you want to do, and you're not thinking about your heart. You don't have to go, oh my gosh, I forgot to make my heart contract. It's going to contract automatically, and it's going to be adjusted automatically based upon your activity. And that takes us to our third type of muscle tissue, which is skeletal muscle tissue. So when we take a look at skeletal muscle tissue, this is the muscle tissue we're going to be focusing on for most of the rest of this PowerPoint, because skeletal muscle tissue is the muscle tissue that comprises the muscles that are attached to our bones. When we take a look at the fibers of skeletal muscle tissues, we see that those fibers are long cylinders with tapering ends. So they're not pointed, they just kind of taper down on the ends. Here we can see an artist's conception of a typical skeletal muscle fiber. This doesn't include the ends where the taper would be. It's kind of cut off there. We can see that this skeletal muscle fiber, one of the differences with cardiac and smooth muscle tissue is that the fiber is multinucleated. There can be several nuclei in a single muscle fiber. There are also dark striations that we see here. So it is striated. It does have cross bands, just like cardiac muscle tissue does. So in that respect, it's similar to cardiac muscle tissue. Skeletal muscle tissue, though, unlike cardiac and smooth muscle tissue, is under voluntary control. We decide when the skeletal muscle tissue is going to contract. Whenever we want to move, whenever we want to get up and walk around, whenever we want to talk, so on and so forth. That's all under voluntary control. We're deciding. Here we can see a micrograph of typical skeletal muscle fibers. They tend to be long. Skeletal muscle fibers can be as long as, there are some up to about 3 feet in length in your body, making up some of your skeletal muscles. So they can be fairly long as well. And we can see here's a long cylinder of an individual muscle fiber, and we can see the cross bands in the fiber, and we can see the nuclei here in purple. They're associated with the fiber as well. Okay, so here's a little chart, and here's what I want you to do. I want you to put the video on pause for a minute. I want you to take a look at this chart, and I want you to try to do a comparison of the 3 types of muscle tissues by looking at each characteristic and deciding whether that characteristic pertains to skeletal, cardiac, and or smooth muscle tissue. So for example, if you look at voluntarily controlled, if you think skeletal muscle tissue is voluntarily controlled, place an X or a check mark in the box in the column underneath skeletal. If you think cardiac muscle tissue is voluntarily controlled, then you'd place a check mark there as well. So there are some of these characteristics that can be demonstrated by more than one type of muscle tissue. But take a minute now and see what you come up with. Okay, let's see how you did. So let's take a look at the characteristic of voluntarily controlled. Skeletal muscle tissue, did you come up with that one? Skeletal muscle tissue is the only one of the 3 that is voluntarily controlled. How about involuntarily controlled? Well, we find that both cardiac and smooth, we mentioned, are involuntarily controlled. Had striations or crossbands. Which two had striations or crossbands? That was skeletal and cardiac muscle tissue. Multinucleated, that was skeletal muscle tissue only. Found attached to the bones, skeletal muscle tissue. Fibers are spindle shaped. Remember, that was a characteristic of smooth muscle tissue. While smooth muscle tissue is pointed at the ends, remember, skeletal muscle tissue is only tapered at the ends. So smooth muscle tissue, because it's pointed, is considered a spindle. Skeletal muscle tissues, because their ends are tapered, not considered a spindle, but more considered to be cylindrical. Which ones have a single nucleus in each cell? That was characteristic of both cardiac and smooth. Makes up the walls of the heart, cardiac, ac pertaining to heart. Fibers are bifurcated. You pick up on that whenever I mentioned that bifurcation means branched. So that would be a characteristic of cardiac muscle tissue. Found in the walls of the stomach. Well, the stomach is a visceral organ found inside the abdominopelvic cavity, and so the stomach we find predominantly smooth muscle tissue in the stomach. And then contains intercalated discs. Remember, those are those desmosomes that kind of stitched them together, and those intercalated discs are found in cardiac muscle tissue. So how'd you do? I hope you got all of those correct. Okay, moving on now, what we'd like to do is talk about structure, moving into the anatomy phase of our discussion. And we take a look at the structure of a typical skeletal muscle. Okay, so here we have, let's say, a bone. Let's say that could be any type of bone like the humerus. And here we have a muscle that's attached to that bone. What we've done is we kind of cut that muscle in half about midway along its length so that we can look into the inside of the muscle and see what it's made up of. As we take a look at what the muscle is made up of, we see, first of all, that there is a connective tissue covering that wraps each skeletal muscle tissue. This connective tissue covering that we see here that goes all the way around the outside of the muscle tissue is referred to as the epimysium. The epimysium is continuous with a tendon. So the epimysium leads into a tendon. Now, one thing that I want to stress here is that I really like this diagram because it really gives us a good idea of the organization of a typical skeletal muscle. But this gives you the false impression here, and I don't want you to retain that false impression, that this tendon is kind of pasted onto or just kind of stapled on, you might say, glued on to the surface of this bone. That's not the case. Whenever we take a look at tendons, this is trying to show the fibers, and what happens is, you may recall that a bone has an outer fibrous covering on its surface, and that outer fibrous covering is called the periosteum. So what happens is this: if my arm is the bone, then a tendon, rather than being stuck to just the surface of the bone like this, as it appears to indicate in the diagram, what happens is the tendons would give off fibers that wrap completely around. They actually kind of blend in with the fibers of the periosteum, so that tendon actually blends in and completely surrounds the bone. So when the muscle contracts, it's going to pull on the entire bone, not just the surface of the bone. And that's going to give a lot more strength and prevent muscles from easily being torn away from the bone whenever a lot of tension is created during contraction. Okay, so that's just a kind of a side feature that I wanted to mention there, so you make sure you understand that. All right, next up, the next component, underneath the epimysium, we're going to find fascicles. A fascicle is a bundle of muscle fibers. So as we take a look at our muscle over here, our muscle fiber, we can, rather our muscle proper, the organ of muscle, we can see here's a fascicle. That's 1, 2, 3, 4, 5, 6, 7 fascicles that are demonstrated here. Within each one of those fascicles, we can see these kind of light red circular structures. These represent each of the different fibers. So a fascicle is a bundle of many different fibers. Next up, we have the paramecium. The paramecium is a connective tissue membrane that wraps each fascicle. So the fascicles are separated from one another inside the muscle fiber by this thin membrane that wraps around the outside. There it is there, and we can see in this muscle fiber, or this fascicle that's been pulled out, we can see the wrapper around it. That wrapper, whoops, let's go back. I apologize for that. But that wrapper is referred to as the perimecium. So this is actually pointing in this one to the wrapper that's around this fascicle right here. Okay, now, so the epimecium is a bundle of muscle fibers, and we can see here's an individual muscle fiber that's been pulled out of the fascicle partway so that we can see it. Now that muscle fiber is also pulled out from underneath a connective tissue sheath that covers it. So each particular muscle fiber also is inside its own little wrapper. And that wrapper is referred to as the endomecium because it's on the inside and it surrounds the muscle fiber. All right, so what we do is we look at this diagram and we can see this muscle fiber pulled out, but we can see that there's a little white wrapper around each one of these individual muscle fibers, and that little white wrapper is referred to as the endomecium, not to be confused with the perimecium that surrounds the entire bundle of muscle fibers or the entire fascicle. Okay, continuing our look at the anatomy of a muscle fiber, let's move from the outside of the muscle fiber, sort of the gross anatomy, and let's start to take a look at the inside of the muscle fiber and sort of take a look at the microanatomy of the muscle fiber. So muscle fibers are cells, and just like any kind of cell, cells are made up of 3 components, right? The 3 main components of a cell are the plasma membrane, the cytoplasm, and the nucleus. Whenever we take a look at muscle fibers, we refer to the cytoplasm as sarcoplasm. Sarco is a root word that comes from the Greek. It means flesh. And so the muscles are the fleshy part of your body. So the cytoplasm in the fleshy part of your body, or the muscle fibers, is referred to as sarcoplasm. Inside the sarcoplasm, then, there are going to be embedded many different kinds of organelles and other structures. One of those organelles or structures we find are myofibrils. These are individual strands of protein that we find inside the muscle fiber. So the muscle fiber, the cytoplasm, the sarcoplasm is going to be very tightly packed, densely packed, I guess I should say, with these structures called microfibril or myofibrils. Myo, by the way, is a prefix that comes from the Latin, and it means muscle. So anytime you see the term myo or sarco, it's going to have something to do with muscles. Now, within those myofibrils, there are fine strands of protein, and these fine strands of protein are referred to as myofilaments. Okay, so you might recall that whenever you studied the cells and the cell anatomy in general, that there were these tiny fibers that made up kind of the skeleton of the cell, the cytoskeleton. And these fibers were microfilaments. Myofilaments are microfilaments specific to muscle fibers. They're fine strands of protein. Now, when we take a look at these myofilaments, there are 2 different types. The first type are what we refer to as actin. And actin is a thinner. It has a smaller diameter than the second type of filament, which is called myosin, and it has a much thicker diameter than the actin does. So these two filaments are going to be very important. They're going to interact with one another during muscle contraction. That's why we take a look at them now. In addition to myofilaments inside the myofibrils, we also have mitochondria. So over here in this diagram, we can see here's a mitochondrion here, here's a mitochondrion here. You may recognize the mitochondrion because of its rod-shaped structure and that kind of squiggly inner membrane that there is that contains these folds called cristae. So that is a mitochondrion there and there. And of course, I haven't shown it or talked about it, but here is the nucleus of that cell as well. So inside that sarcoplasmic cell, it's going to be densely packed with these myofibrils. And here we can see individual myofibrils sitting inside the cell like canisters. Okay. And I like this diagram a lot, again, but diagrams don't always do complete justice to everything you want to be able to demonstrate. It appears right now as though these canisters are empty, but actually these canisters are filled very densely packed with these myofilaments, the two myofilaments, actin and myosin. Remember that actin is going to be the thinner of the microfilaments, and myosin is the thicker of the myofilaments. Now over here on the side, the artist is trying to show actin and myosin. These thin lines represent actin, and these thicker lines represent myosin. But they give you the impression that they're only wrapped around the outside. These filaments are packed all throughout this fiber. Okay. So now let's go and let's take a look at an individual myofibril. So an individual myofibril is going to contain. Well, let's start out where we're at here. Let me backtrack a little bit. This is a myofibril, or this is a muscle fiber, and this is a myofibril right here. And we can see packed into that myofibril, here are the individual myofilaments. Okay, so we have the myofilaments there, the actin and myosin. Now it's a little difficult up here in this part of the diagram for us to tell the difference between them. So what we're going to do is we're going to blow up this little section of the myofibril, and we're going to make it large so we can see the individual myosin and actin filaments. Before we get into that, though, when we take a look at the myofibril, it's actually divided into sections called sarcomeres. The sarcomere is the functional unit of muscle contraction. What we mean by that is that when a muscle contracts, what happens that allows that muscle to undergo contraction, or what kind of changes take place when that muscle undergoes contraction, are going to occur in the functional unit. In other words, they're going to occur in the sarcomere. So it's changes in the sarcomere that occur that allow for muscle contraction to take place. They're the functional units of muscle contraction. Now, whenever we take a look at sarcomeres, there are many sarcomeres within the myofibril and therefore within the fiber itself. Sarcomeres are separated from one another. We can tell where one begins and the other ends by the position of what are called Z lines. These Z lines are dark lines that separate one sarcomere from another. They're called Z lines because they kind of zigzag back and forth. So if we go over to our artist's conception here, we can see here is a Z line here, and here is a Z line over here. So this area between those two Z lines is referred to as the sarcomere. Now, in the center of the sarcomere, we're going to find our thicker myofilament, and that thicker myofilament is referred to as myosin. So they're positioned in the center of the sarcomere. And then overlapping the myosin, we see these thinner filaments. They partially overlap the myosin filaments, and they extend to where they are anchored to the Z line. So we can see each of the actin filaments, and we can see how the actin filaments are connected to the Z line. Now I got to tell you now that this Z line is not actually a line at all. It's more like a disc. And some anatomists actually refer to it as the Z disc. It's called the Z line because whenever you look at these underneath a microscope, remember the microscope can only see in two dimensions. So it appears to be a line rather than a disc, but it actually is a membrane, and sometimes referred to as the Z membrane, or a disc-shaped structure that separates the sarcomere. It has three-dimensional shape is what I'm trying to say. Okay. So, whenever we take a look at the sarcomere, we can look at some additional components for geography so that we understand where we're at any one particular time. So down here, this is actually a electron micrograph of a section of a myofibril. And this section of the myofibril has 1, 2, 3 sarcomeres in it. And I can tell the sarcomeres because here is a Z line here, or as designated in this diagram, a Z disc. Remember, sometimes they're called Z disc or Z membranes. So this distance from here to here is one sarcomere, Z disc to Z disc. This distance from here to here, another Z disc to Z disc, is another sarcomere. And here's the distance between here and here is a third sarcomere. Now, because there are some variations as we move throughout the sarcomere with regard to density, we refer to different regions of the sarcomere. One of the regions that we have is referred to as the A band. The A bands are dark regions of the sarcomere where the actin and myosin overlap one another. So we can see, remember, the actin extends from the I band to partially overlap the myosin. So we can see here these dark areas right here. This is, if we think back to that artist's conception, where the actin is overlapping the myosin partially at. On this side, and over here, you can see it's lighter here, and then it gets darker where the actin is partially overlapping the myosin. Now, it doesn't completely overlap the myosin, so you can see it gets a little bit lighter here. Okay, so this area where we have the overlap is referred to then as the A band. In addition to the A band, you also have I bands. I bands are light areas of the sarcomere that consist of actin only. So we can see in this area right here, this is where the actin is attaching to the Z band at, or the Z disc, I should say, and it's only actin, so it's much lighter because actin is much thinner than myosin is. So this is an area from here to here that's referred to as the I band. So we have alternating dark A band, then a light I band, then a dark A band, then a light I band, and this gives us our kind of cross-banded nature that we see in both skeletal and cardiac muscle tissue. Now, in addition to the A bands and the I bands, we also have designated here an M line. This M line is the dark line at the center of each A band. It consists of myosin only, but if we think again back to that artist's conception, take a look back at that slide, the artist shows the myosin has kind of a bulging, thick area right in the center of it. And that's what we're seeing here in this micrograph, where the myosin is much thicker in the center, so it's a lot darker right there. So we can see that's the M line. Here, the myosin's a little bit thinner, so it's not quite as dark. Here, the myosin and actin overlap, so it's darker. And then we get lighter again because this is only the actin, where it extends out and attaches to the Z disc. Okay, so looking at those regions of the myofibril and within the sarcomere itself. Okay, we talked about some of the organs, or organelles, I should say, that are inside the cell. One of the organelles we haven't talked about yet, and it's very important to muscle contraction, is the sarcoplasmic reticulum. The sarcoplasmic reticulum consists of a network of interlocking tubules, small tubes, that form a network over the sarcomere. So if we take a look at this artist's conception here we can see a network of tubules that's kind of lying over. Look, here's the region where we have the actin and myosin. So this network of tubules that we have is referred to as the sarcoplasmic reticulum. Now, near the eye bands, the tubules, that's near the edges of each sarcomere, right? Near the edges of each sarcomere, the eye bands, the tubules are going to fuse with one another. We can see how they're coming together and they're fusing right along here. And they form these kind of end sac regions. These end sac regions are referred to as cisternae. The cisternae, if we look over here on the right side of the diagram, are going to occur in pairs. So here's a fusion of tubules here on this side, and this is referred to as a cistern. And here we can see another fusion of tubules over here, and this is referred to as another cistern. Together, plural cisternae, these end sacs. The function of these end sacs is to store calcium inside the sarcoplasmic reticulum until it's going to be released during a muscle contraction. Now, one of the things I failed to mention here was we have a cistern on this side, we have a cistern on this side, and then we have this kind of long tube that runs kind of between them. This long tube that runs between the two cisterns is referred to as a T-tubule. So T-tubules are a system of tubes that run perpendicular, so the sarcoplasmic reticulum is running horizontally this way and ends in these end sacs called cisternae, and then there's perpendicular to the way the sarcoplasmic reticulum is running, we see these perpendicular structures which are referred to as T-tubules. These T-tubules are very important. They're going to open to the surface of the muscle fiber. And remember, a muscle fiber is a cell, so the surface is going to be the plasma membrane. The plasma membrane of a muscle fiber is referred to as the sarcolemma. Remember, an alternative name for the plasma membrane is the plasmalemma. And so here, we're just designating that this is a muscle fiber or a muscle cell, and we're using that Greek word sarco, meaning flesh. So this is the sarcolemma, the plasma membrane of the muscle fiber. Now, if we take the T-tubule here in the center and the two cisterns on either side of the T-tubule, together they make up what is referred to as the calcium triad. So we have the sarcoplasmic reticulum cistern here, the cistern here for the sarcoplasmic reticulum on this side, and the T-tubule in the middle. Together, these three make up a structure called the calcium triad. This is going to be very important to understanding muscle contraction. Okay, and with that in mind, we come to the close of our first lecture PowerPoint on the muscular system, and we've taken a look at the anatomy of skeletal muscles. So now we know that what skeletal muscles do. They undergo contraction. That creates pulling forces that pull on bones to create movement, and take a look at those other functions that we saw as well. They're also responsible for those. But now the question becomes, how does muscle contraction take place? Okay, what happens when a muscle contraction takes place? That's called the physiology of the muscle contraction. How does it do what it does? How does the muscle contract? We're going to stop here, and in our next PowerPoint, we'll pick up with that look at the physiology of muscle contraction.